Hello students, welcome to LEELA'S tutorial. So in last class we have discussed about batch cultures. Today we will discuss continuous cultures. So why we have to go for this batch or continuous cultures? For the production of secondary metabolites. What are secondary metabolites? These are pharmaceutically bioactive compounds which are produced by the plants. So what are these plants using these secondary metabolites? No, plant doesn't use secondary metabolites neither for their growth nor for the reproduction, right? So why they are producing this? They are producing these in order to protect them from the external environment, right? Such as other insects or any other invading organisms, right? So here these secondary metabolites play a very important role in pharmaceutical industries, cosmetic industries, agriculture industries and all. So here we want those products to be produced in bulk. So we can't keep on growing the plants for so many years in order to extract those compounds from the plants. So what we do, we make use of our plant tissue culture, right? Where we are culturing the cells and allowing the cells to produce that particular byproducts, right? So let us see how we are going to perform. First, it is the selection of the plant. The plant which is having the medicinal ability, you are going to select, and then you are going to check which part of the plant is producing that particular secondary metabolite. Either it is a leaf or the flower or it is a root, right? So based on that, you are going to select the explant and inoculate in plant tissue culture media, and in presence of auxins and cytokinins, 1:1 ratio, the callus will be induced. So once the callus is undifferentiated mass of cells. So undifferentiated mass of cells, it is a group of cells. So what you do, you disaggregate the cells and form a single cell suspension, right? So now you have the single cells which can be used for your continuous cultures, right? Otherwise, you can make use of immobilized cells. Immobilized cells are also referred as encapsulated cells or entrapped cells within a gel matrix, right? So here you are making use of calcium alginate gel so that they are immobilized inside that gel, right? You can go through that immobilization process or encapsulation process complete in detail in the previous classes, okay? So here once you get the beads, immobilized beads, they protect these cells from the damage, okay? Whereas here, when you use these naked cells or when you use these disaggregated cells in the culture process, the cell wall of the cells may get ruptured and they cannot be reused, okay? So once you are making use of this, any one of these, perform the continuous culture process where continuously the media will be added, right? The cells will be growing and producing the secondary metabolites continuously, and again the media and the secondary metabolites will be collected continuously, okay? And the secondary metabolites will be sent for the downstream process for purification continuously, and the used up media will be replaced by the fresh media continuously. So here everything is continuous. They are not stopping this process. Initially, see here, you can see first it is the continuous bioreactor which consists of 2 tanks, whereas in case of batch culture or fed batch culture, it is only the single tank, right? So here the continuous culture consists of 2 bioreactors. One consists of sterilized media and other one the process for your continuous culture. So here you are going to sterilize the tank, then add the media up to the working level, add the immobilized cells, okay? Then start the process by maintaining the internal environment such as temperature, pH, nutrition for the cells to grow and produce the secondary metabolites. So once the process has been started, what you do, you start releasing or letting in the fresh media slowly. It is very slow passage of the fresh media, and here after completion of the process after certain time, slowly you will be letting out the spent media. So that means here continuously media is passing and also continuously the secondary metabolites and the used up media is letting out. Okay, so it is very slow, such that by the time the media passes through the outlet, the cells are producing the secondary metabolites, and the secondary metabolites along with the used up media, it will be collected from the outlet. So how they are going to check whether the secondary metabolites are produced or not? By means of photocell. Right, here they will be passing the light, and there the light detector will be checking the turbidity. Okay, so and here you have the agitator, as we have discussed in batch reactor. Agitator or impeller, it will be rotating, which is connected to the motor, so that it will be mixing up all the fresh media with the immobilized cells. Right, and then you have the baffles in order to avoid the vertex formation. And if there is any increase in the temperature or the pH, here you have the pH probe, so that it will be adding acid or base. And then you have the temperature probe. If there is increase in temperature, the cooling jackets, the cool water will be sent. Right, so cooling jacket or the thermal jackets will maintain the temperature. It will absorb the excess heat. Okay, this cooling jacket is present outer surface of the fermenter or the bioreactor. Right, baffles are present inside in order to avoid the vertex formation. So these are the baffles, they avoid the vertex formation because it is agitating or the impeller is rotating. Right, so here the rotation is also very slow, so that no cells will be damaged. Okay, so here you will be adding media continuously, and continuously you are collecting, and this process will be carried out for several months. Okay, till when it will be carried? If there is no any disturbance or any contamination, the process will occur for several months to year. Right, so here if you take a graphical representation of this continuous culture, here you are seeing number of cells in the y-axis and time in the x-axis. Right, so initially you have added the cells in the bioreactor. Right, so it is the number of cells is same, stagnant. Okay, the number of cells for certain period of time, there is no increase in number. The cells are, if you are adding 10 cells, only 10 cells have been maintained. That means the lag phase is the adjusting phase. Right, the cells are getting adjusted to the new environment. Right, once they get adjusted, they start utilizing the nutrients present in the media, and they start growing. This is referred as log phase, where the cells are growing at exponential. Right, you call it as exponential growth or 2 to the power n growth. Right, so here the exponential growth is observed. So once the media or the nutrients are depleting, what it will do? It will start production of secondary metabolites. So cell number has been increased, and also cell growth or the increase in size of the cell has been occurred. After that, it is the production of secondary metabolites. When the secondary metabolites are starting, right, when the production of secondary metabolite is initiated, what you do? You will not allow the nutrients in the media to get depleted. Right, so what you do? You slowly add the media along with the nutrients. You open this knob and slow inlet of the media will be there. Right, so there will be slow passage of the media and slow outlet. So that continuously here the cells will be doing their work, producing the secondary metabolites. So secondary metabolites will be collected. Okay, secondary metabolites will be collected slowly. By the time fresh media comes, again the cells will produce secondary metabolites, and secondary metabolites along with the spent media will be out. Okay, so then from here again purification step occurs. So here this is the, see here in the growth curve there is no decline phase. In batch culture, how we have shown the curve? Lag phase, log phase and stationary phase, where the secondary metabolites are produced. After depletion of the media, they are entering into the decline phase. Okay, after that you will be stopping the process and sterilizing the fermenter and again start new batch. Right, whereas in case of fed batch, this is in case of batch, in case of fed batch fermentation, what you have done? Lag phase, log phase, stationary phase. Right, when the stationary phase production is lowering, you will again fed the fresh media, again stationary, again you add little bit fresh media, again stationary. This is your fed batch, right? Whereas here in case of continuous culture, you can see the production of secondary metabolite is increasing continuously. Okay, so that is the difference between your batch, fed batch and continuous. If you have any doubts, put it in comments. Subscribe for further videos.